This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hey everyone, welcome to Audiommunity. This is episode 13. I'm Kevin Bonham, and joining me is Kate Franz.
Hello.
And Matt Woodruff.
Hello again.
And we've been kind of on a hiatus for a little bit. Matt managed to record and post at least one mini-sode, although it didn't have any of the intro and outro music.
No.
Because that's my job. And I was gone on a honeymoon and decided that I didn't care about this podcast for at least a little while.
Walk that back.
Had a stronger interest in other parts of the world.
There you go. Okay. Yeah, I dig that. I was actually just going through photos. I took over 1,000 photos on the trip.
It doesn't surprise me one bit.
3.5 weeks. Yeah. And some of them are very nice. It's fun going back through and seeing pictures of Siena and wishing I was there again. But anyway.
I went to Mexico and took 6 pictures, I think.
Were they? How many of them were of you?
2.
Okay. I feel like that's a reasonable ratio.
33%.
Yeah, that's a reasonable ratio.
I think probably less than 1% of the photos taken on the honeymoon were of me or had me in them.
Novartis has a complex in Siena.
Oh, really?
Yeah, they do vaccine work over there, or they used to do vaccine work before they decided that they didn't want to do any vaccine work anymore, sold the entire thing to GSK in return for, I think it was cancer immunology. Anyway, okay. GSK might now have a center in Siena, or now they do cancer stuff. Anyway.
Cool. Well, I don't want to live in Siena.
No.
But it's a very nice town. I quite enjoyed it, and Tuscany is awesome. But anyway, the point of this episode is going to be sort of a slight departure, maybe, from our normal topic. I mean, we're going to certainly touch on immunology, I think, but we got an email from a listener, and his name is Bruce. I don't know if we want to do last names.
No.
Or if these people care.
No.
But Bruce writes, "Hey, doctors and Kate." As an aside, I really love that other people also can fit on Kate.
I know, I was like, wow, just like state your allegiance to Kevin right off the bat.
Anyway, he says, "I'm tired of trying to design this independent study and thought I would send you an email with some questions and suggestions. First off, it's sunny 20 degrees Celsius with 54 kilometer per hour winds here in the Great White North, a.k.a. Lethbridge, Alberta." This was in May. I don't know why it was still white there.
Because it's freezing and it's awful, and it's not, I don't know anything about it, but do you remember
This
April,
is why
though?
he has allegiance to Kevin.
Right.
No. But do you remember April? April was still freezing cold. It was like 40
That's
degrees
true.
in April, so
Yeah.
I'm not surprised it was still freezing and snowed over in other places in May.
That's true. We drove through a blizzard in the Alps in like late May. It was kind of crazy. Anyway, so he gives us the weather report because, of course, he listens to the TWIV Network podcasts, and they always start with weather. We, at one point, had thought about doing it, but thought that it was too much of a riff on there, or too much of a rip-off of their podcasts. Anyway.
So we started doing drinks, right?
We started doing drinks. Speaking of which...
Yeah, I'm drinking coffee today, which is the first time ever on this podcast, because I think we generally don't record in the mornings. Usually it's well past noon when I start drinking scotch.
It's true. It is. It's currently morning, and I'm drinking tea, PG Tips, which is quite tasty, and Matt seems to have just disappeared.
And reappeared. Yay!
Yeah, blacked out.
Anyway, okay, so moving on. Bruce continues. This is really bad. If we do this every time we get an email, it's going to be a nightmare. So he says, Bruce says, "Second, because I've been listening to This Week in Parasitism, one of those TWIV Network podcasts, I was wondering if you guys could cover some immunology of a eukaryotic infection. Yes, we should do that, although I know nothing about it. We should find some papers.
We'll figure it out. It has something to do with eosinophils, which we've already established we don't know anything about, but we can certainly try.
But neither does anyone else.
Right.
So he says, "I understand that eosinophils play a role in parasitic infections, and I think you even mentioned the body's method for fighting large parasites is to create an inhospitable environment around whatever critter is invading, because obviously phagocytosis is not in the cards. That is true. As a nursing student, I should know that an elevated eosinophil count is indicative of a parasitic infection, but sadly that's about as deep as my understanding goes." Yeah, yours and everybody else's, Bruce. Don't worry about it. Eosinophils are...
We don't get it.
Yeah, we don't get it. Third and finally, why can't my immune system pull its head out of its ass and figure out rhinovirus? I'm on my 3rd cold of the year, and it's getting getting old. I assume it has something to do with antigen variation among rhinovirus. I know that I could simply Google it and find the answer in 10 seconds, but that would take far too much effort, and I'm sure you appreciate a good excuse to get distracted from your work. I mean, yeah, you basically have it. Rhinovirus is better at evolving than our immune system. Or rather, it's...
No, that's not the only thing. It's that there's hundreds of viruses that can give you the cold. So you have like adenoviruses, rhinoviruses, like there are all these different, there's a whole bunch of viruses. So you might be getting a different virus every time. So there's diversity within the types of viruses. So there's like different subtypes. And so you could be getting different types of rhinoviruses, but then you also could be getting a whole nother virus in general. So you're just never going to catch up.
Yeah, it turns out that infecting airways is a really, really good strategy for viruses because it's really easy to transmit. It's really sort of accessible because you're constantly breathing. So lots of viruses have figured out that infecting airways is a really great strategy for surviving. Okay, moving on. Okay, so I have a few more questions. Is the Janeway textbook my best bet for an immunology book?
And I think I responded to that. I said that you can't go wrong with Janeway. We both worked with Shiv Pillai while we were at Harvard teaching undergrad courses, and I actually really liked their book. And I can't even remember the name, but I did send you the recommendation.
It's Abula Boss,
Shiv
Pillai, and Andy Lichtman. So I think it's like immunobiology or something like that. But it's
really good.
Immunobiology is Janeway.
Yeah.
But there's also another textbook, I think. But yeah, I mean, Janeway is sort of the de facto standard. A Boss is really good as well. And I'm actually working with Shiv to develop a sort of online version for HMS, for Harvard Medical School, like a online course that it sort of follows his textbook as well. So yeah, but Janeway's great. And of course, I have some allegiance to Janeway because my boss's boss's boss.
It's because of this vague connection.
This very tenuous connection that actually means nothing.
It doesn't mean anything, but whatever. Okay, so then he goes on to say, I notice you guys often talk about ethical implications of some research in your podcasts. My independent study is for a fine arts and humanities elective, and I have decided somewhat to focus on research and or clinical ethics. As a preliminary study, I have started reading Integrity in Scientific Research, Creating an Environment that Promotes Responsible Conduct, published by the Institute of Medicine, National Research Council. I have not read much yet, but I had a creepy Joseph Mengele-ish idea, and I'm going to preface what he actually says by saying, yeah, it's super creepy, but I think starts an interesting ethical conversation that I will probably be on a different side than Kate and Matt on. But
I don't think so.
That's what I'm
actually
going on. Please don't judge, because I know it's right muffed. Is that a Britishism? I know it's right muffed and probably impractical. Oh, no, he's from Canada. And probably impractical. According to Taber's Medical Dictionary, about 11 in 100,000 births are, I don't know if that's 11 in 10,000. The comma's in a weird place. Anyway, are anencephalic, meaning the child lacks cerebral hemispheres or more. The condition is incompatible with life, and most anencephalics are deaf, blind, and unable to feel pain, according to Wikipedia at least. Would these not be perfect models for human immunology research? Assuming they can be kept alive with an artificial respirator, which would probably cause immune complications, there are definitely a lot of ethical issues with such a proposal, along with monetary issues, etc. Nonetheless, an interesting thought. Or am I totally crazy and should never mention it again to avoid being labeled as such? The answer to that last is you should probably avoid mentioning it unless you are
at least in mixed
a
company.
Room full of philosophers. Yeah, exactly. I wouldn't spread it around unless it's clear that it's hypothetical. Anyways, I thought I would shoot you an email so y'all know I appreciate what you're doing to supplement my, and then period. I think there was supposed to be something else there. I wish you all the best and happy Friday. A month and a half ago. Congrats on the marriage. I think that's to me, but maybe to Matt, because Matt's also getting married this year. Kate, please don't take offense from my salutation.
I believe that I'm also getting married. Thank you.
Oh, right. I forgot about that.
Thanks, Kevin.
Sorry. Have I gotten my invitation yet?
No, I haven't sent out anything. I don't even have a venue. I'm very bad at I have a dress.
Okay.
But
well, okay,
I have nothing else.
Taking care of the important things. Good. Okay. So this idea that you would take babies that are born that lack cerebral hemispheres, they're deaf, blind, and unable to feel pain, maybe. I mean, according to Wikipedia, I don't know. But let's just say hypothetically, if we could know that they didn't feel pain, would this be a viable research subject? And this kind of will segue nicely into what we're actually going to talk about, which is some research that was done on human embryos that were going to be not viable. So these are sort of at the single cell stage stage or multi-cell stage. So we'll get to that in a bit. But what do you guys think?
So why don't we back it off even a little bit more than that? Let's start at, I think, the simplest ethical boundary, which is, let's assume that you could have a baby that was born, or whatever you are going to call it, a human that is born without a brain, totally without a brain, right? So you can keep it alive solely through breathing tubes, solely through, obviously you need some brainstem function in order to make the thing's heart pump. But if it has absolutely nothing as far as we now define life, right? Because we now define life by brain activity, right? So brain death is sort of the ceasing of life in our current medical understanding. So if the human that's born is never alive due to a lack of brain, I guess you could pretty easily make the argument that it was never human to begin with, because that is how we're defining the life of a human, no?
Right. I mean, sort of. So
maybe you could define that, but I don't think a lot of people would agree with you.
So you
could set up that definition, but I think that if you say a baby that's born stillborn is not human, I don't think you're going to get many people to agree with you.
Without a brain.
I mean, if you're born without a brain, you're being born dead. And you just said because life begins at birth.
But it was never alive to begin with, because it didn't have a brain or brain function.
Well, it was never, but it might have been alive, but just not. So I think maybe we need to step back even further and say, what are the principles, the ethical principles around which we do research? Because I think, most people will agree pretty emphatically that doing research on bacteria or on single cells is ethical. Mm-hmm. And I think most people would say, at least nowadays, that experimenting on normal human beings without their consent, or doing a lot of the sort of biological research that we do, most people would say that's unethical. Mm-hmm. And so where in between those two boundaries are we going to draw a line, if we're going to draw a line? And I think that sort of thinking about the principles that we use to make the decision about whether something is ethical or not is important, because some people think animal research, doing work on mice, is unethical. Mm-hmm. I think most of those people would say that doing work on worms, like a C. elegans, is probably okay, maybe, I don't know, or fruit flies. I think that different people are going to draw boundaries in different places. Mm-hmm. So for me, my version of morality is taken a lot from Sam Harris, who wrote a book called The Moral Landscape. And the idea there is that essentially morality is defined by conscious creatures. And essentially, if you are doing things to harm conscious creatures, that's unethical. If you are doing things to promote the well-being of conscious creatures, that is more ethical. And so there's sort of a line. And so the idea there is that mice are less conscious than humans. Worms are less conscious than mice. So there's sort of a moral,
spectrum.
There's a number line.
Like a rheostat.
Right. Oh my God, don't start with that. That rheostat thing is an inside joke. Anyway,
sorry.
It's okay. It's from first year of grad school, and it was I hadn't thought about that in a long time. Anyway, yes, there's a moral rheostat, and it's defined by conscious creatures. And so, on the one hand, I would say if you have an organism that is not conscious in any way and could never be conscious, and you are doing experiments on that organism, that is morally equivalent in some ways to doing work on tissue culture, on cells in a dish. But I think that you also have to consider the well-being of the people doing the research and the implications of that. So if you had an organism that looked exactly like a human infant, and you have researchers that are doing experiments on this creature, even if that creature is not suffering in any way because it's not conscious, it has no ability to suffer, the researchers doing the experiments, I think, might suffer some sort of harm mentally, cognitively, from doing this research on a thing that looks human. Or there might be implications in terms of those people's ability to then differentiate and say, like, well, if this is okay, what about, infants are barely conscious, like actual human infants are barely conscious the way that we would define it, so maybe that's okay. And so there's sort of a slippery slope there that is a little bit more ambiguous, in my opinion.
Well, I think with the infants, it's I think the argument there is like, it's not that they aren't conscious at the time, it's that they have the ability to develop it.
Yes.
Right.
That's true. So there might be lasting harm that will affect them when they become conscious.
Yeah.
Right. Mm-hmm. I spoke with my fiance about this briefly last night, and she came down on the presence or absence of a face. And I think this is where most people will actually get caught up in this argument, is things that we strongly identify human and not human based on visual cues. I mean, we're designed to do that. And I think it would be, as you mentioned, really difficult on the researchers, actually, and on the perception of the entire thing, which is actually probably why I think it would be problematic from a societal level, not so much from a harm of the thing you're doing the research on. I think it's a dangerous game to have society who are going to be overwhelmingly identifying with the research subject as something that looks human and looks like a normal conscious living thing.
Right.
I think that there's a real problem with the societal implications of knowing that that sort of thing's going on without the luxury of having explored this from a philosophical level, I think, in the way that by sitting around talking about this, we will have explored it.
Right. And so I think that that's an important distinction, is that, ethically, what responsibility do we have towards the research subject? And if it doesn't have consciousness, if it doesn't have the ability to suffer, I don't think we have a moral responsibility towards an organism that has no ability to ever gain consciousness.
You would have to also stipulate that everyone around that organism, right? The parents, things like that, everyone would have had to have signed off on this. Everyone would have, this would be a donation to science sort of thing, right?
Yeah,
that everyone has signed off on.
So
that's a separate question.
Right.
I want to talk about something. So you're saying that in the case of an infant that's born without consciousness, you should be able to do research on them.
I'm not quite saying that. I'm saying that the moral implications of that sort of research are not moral implications directed at the organism, directed at the subject of the research. I think the moral implications are sort of one step removed. The researchers, the family of the child that was born with this condition, all of those things, I think, are real moral concerns that we can't ignore. And I think that probably, even if that organism that has no consciousness and can't suffer, even if that organism is a valid subject for research from that organism's perspective, I think that probably, on balance, that research shouldn't be done because of these sort of implications, the moral implications to the family and to society.
My issue with this is the way that so in the email that this was set up was that I took it as these anencephalic infants will be essentially incubators. You have an organ system, so you can use these as incubators and as a research tool. And my whole issue with this is this might be a little bit more concrete than how we're talking about this in the abstract, but that's an awful, this is just bad science. We don't do that because it's just bad science. You have
so that's
one immune system that you're looking at. The point of science, the point of why we use model systems is that we can get high numbers and reproducibility of the experiment under the same genetic background, under the same environmental backgrounds. So taking a 1 in 1,000, 1 in 10,000 baby and using them as an incubator, even though I would find that morally abhorrent, it is also just bad science. It's why twin studies aren't good. Even though they're a cool, interesting idea, you only have 2 people. So even though you control for genetics, you can't actually reproduce anything.
Right. Yeah, that's an important point. So I think that you could also make an argument that in this particular case, so I said that on balance, this is probably not a good thing morally. I think you could make the argument that if this tool was so good that you could anticipate making enormous and important discoveries that would save lives, that from a moral perspective might outweigh the suffering of the researchers and the family around the use of these things. But I totally agree with you. This is not really that great a model system. I don't think you don't gain much, if anything, over the models that we have that we're morally comfortable with.
And even to address it more directly, I think the anencephalic model is very bad, just because these babies would be dying and they don't have brainstem activity. I'm not sure, they die in minutes to hours.
Right.
And Grey's Anatomy. But
always got to bring it back to Grey's Anatomy. Right. Damn it, Kate.
We've talked.
But I will say the sort of situation that happens in these cases is that you have tissue donations. Children who die very young, tissue donation is definitely an issue that gets brought up. And those sorts of situations, the research is worthwhile. Like there can be like very valuable tissue that people, wait to acquire over time. And so like the argument that like it's not scientifically sound is not always the case. But if you're looking at the one like incubator baby situation, that is bad science. But when you go to like the tissue donation and using that like for research, I would say it's a slightly different situation.
So let me expand on this a little bit. So let's say that in 5, 10, 15 years, we develop the technology to take embryos that are discarded from IVF that aren't going to ever be implanted. And we have a way that we can separate out an individual embryo. We can like let it develop to a blastocyst. We can separate that out into hundreds of genetically identical copies. And then we can induce them to develop into an infant without a brain. So we can make genetically identical multiple copy number incubators, as Kate said. That would be a useful research tool. You could do good science with high reproducibility. At that stage, let's say we could use this as a good research tool. We could sort of get rid of, and I think this idea of thinking about the implications of if this was a good tool, this will come up later in our CRISPR talk, but if we could make it a good tool, would that be ethical?
No.
And I still think the answer is probably no.
No.
If it looks like a human, even again, I don't think that those.
I know you're playing like the other side to it. I think just to be clear, like I don't think Kevin is saying that this is a good idea.
No, I'm saying even if we could make it a good tool, I still don't think morally, if it looked like a human,
I think the issue there now is like you are making a human and you're removing its consent because you have it.
Exactly.
You are making it without any ability to think. And so like now we're into the issue of a vulnerable population that's unable to consent to human research. So like one of the number one things in human research is that the subject needs to be completely informed and able to give consent, which is legally why we don't do research on prisoners, because they are unable legally to give consent.
Right. So, but what about stem cell research? I mean, if I think that there's not a huge difference between something that has been programmed from the blastocyst stage to never develop consciousness. It's sort of this gets back to my definition of morality that is dependent on conscious creatures. And so if that creature was never going to be able to be conscious.
But it's a little bit different. I really do think that there's a difference between a blastocyst and a full developed human infant.
Right. But we're not talking about a full developed human infant. We're talking about a.
Oh, I thought you're still putting it against like the weird matrix babies that you'd make.
Well, he's saying that you could interfere early, right, in the blastocyst stage,
right. So they would never develop a central nervous system, right.
So there's something, and I don't know quite where it is, but something about that interference. Yeah, something about that interference rings really uncomfortable to me. And I think it's the idea, I can't put my finger on it, really. It's the idea that we're intentionally creating a thing that had the ability to be conscious, and we are selectively, I think Kate, you pointed it out, it had the ability to have consciousness at some point, but what we're doing is selectively subtracting the ability to consent and the ability to be what it could have been. And there's something about that it just
it's a tough question, but I don't think the morality is super clear, because, and this is a sort of weird thing to do in moral discussions, which is to do the, reductio ad absurdum, the sort of taking it to its most extreme case. And I think, the line between stem cell research and the sort of research that I'm talking about is not super clear. Like, what if you took an embryo, again, these are embryos that are never going to be implanted, so in the absence of the research that we're doing, they're going to be discarded.
Yeah.
And you use that blastocyst to develop a heart, a fully functioning heart, and do research on that. Mm-hmm. I think you guys would be comfortable with that, right?
Yeah, I think so.
Or if you use it to develop some other organ system, probably not a brain, because there's something weird about brains, but...
Well, I think here's is there any other way to do this? I guess would be my question. Is this the only way to do this research? Is to use a human embryo? And I would argue that the answer is no. There are other ways to do it. Now we have induced stem cells. So maybe actually I will change my answer that like the concept of it, I think I'm okay with, but only if there were no other way and the research was actually worthwhile.
Yeah, I think those are important caveats. You gotta weigh the morality. I would love it if we could never do mouse work. I think there is something about I think there's a level of moral hazard with doing research with mice. I think that on balance, mouse research is a positive. I think that it is morally acceptable to do animal research because we can do science that we wouldn't be able to do otherwise, and mice are
have successfully done that. Yeah,
certainly.
Right.
If at some point we could eliminate all animal research and only use artificial systems, I think that would be great, or computational models or something. That would be great, but we're not there.
Could I make
Oh,
sorry, go ahead.
No, that was it.
Oh, okay. Basically. I was going to make another. I want to actually go really quickly, touch back on like our conversation of like the incubator system. I do want to say another reason why, like what you were saying, how you could make, if you made like some sort of organism that you subtracted the consciousness out of,
you prevented consciousness
You prevented it from developing. I still think that we would never do it because it'd be so expensive and it wouldn't be a good system, because you could do that in animal systems that are very close to humans. Like you could do this in like chimpanzees or like monkeys, and we don't, at least as far as I know. And so I'd also like to put that out there, that like it's just not a good idea scientifically, again, because you could do something very close, and as far as I know, it's not happening.
Right.
Or we could take these systems that we're more comfortable with and start putting in human components, right? With something like the CRISPR-Cas system, which is what we're actually going to be talking about. You know, if we use these systems we're more comfortable with the technology that we're now capable of utilizing, you know, we may never, this is a moral hazard we might not ever have to wade into.
People have freaked out about the use of humanized mice and misunderstanding around what that system entails.
Right.
But anyway, let's move on to our actual discussion. So some of our listeners have probably heard of CRISPR-Cas, which is this new genome editing technique. There was a really good episode of Radiolab recently where they sort of talked about this. This always seems to happen. We always come up with ideas for podcasts and then wait too long to do them, and then somebody else does them. Well, so this was the case. We had talked about doing a CRISPR episode.
This is without annoying sound effects. Radiolab gives me a headache. I can never listen to it because I get too disoriented and my head starts to hurt.
So the idea here basically is that we now have a tool that we took from bacteria that essentially allows us to go in and fairly precisely remove or add genes from a genome.
Right. Removal is much easier, by the way. Much easier to remove.
I also was going to take issue with precise.
Right.
I said relatively precise.
Yes, more precise than in the past.
To the extent that we have not been able to do this in the past. And so we've talked before on this podcast about knockout mice. This is a technology that's been around for decades, where you can basically take a mouse embryo and you can use the underlying DNA repair machinery to knock out or knock in genes. And we've used this to great effect in mice, but this technology doesn't really work for differentiated cells. So to use this on, say, human cell lines, or in a number of circumstances, this technology was not usable. But CRISPR-Cas, we can use this system to actually alter human cells, either stem cells or tissue culture cells, and we can do some genome editing. And so this technology has sort of exploded into biological research. The ideas have been sort of filtering around for a while, but it was only a couple years ago that a paper was published showing that this system can be used as a genome engineering tool, and adoption has been rapid, and a lot of biologists are really excited about the sorts of research that we can do with this tool.
Right. So it's worth pointing out why that excitement has been warranted, I think, in the biological community. And so you mentioned these knockout mice, these transgenic mice that we've been making for a long time. And so we have been making them for a while, but the actual physical process of making these mice is not so easy in a traditional sense. So basically what you're doing is you're going through this really slow process of multiple generations of mice being altered and then back crossed. And then basically at the end of the day, if you're going to traditionally make a transgenic mouse, you're talking about 9 months to a year and a half of breeding that mice. So when you get an idea for experiment and you say, I really want a mouse with this specific knockout, you can do that experiment, but you can do it between 9 months and a year and a half from now, when all that breeding comes to conclusion. The real breakthrough that CRISPR-Cas has done in whole organism systems is that you can go into an embryo, into a blastocyst, and essentially at the first generation get mice that display these traits, right? So you basically dropped the time frame from the 9 months to a year and a half down to, if you've got a really good system worked out and you've got people that really know the technology, you can get it down to like 3-4 months on the first generation. And that's huge.
And also, it's also expanding the range of organisms that we can do this in.
Yeah.
So this organism, or this method that I talked about before, it works in mice. It doesn't work in rats. It didn't work in, say, monkeys that we use for HIV research, for example. And again, it didn't work on human cell lines. And so CRISPR-Cas seems to be far more extensible. It works in just about every organism, maybe every organism that it's been tried in. I don't know if there are examples where it's failed. So this is a really broad system. And we should also say that the ability to knock out genes is an incredibly powerful tool for understanding how things function. So being able to remove a gene and replace it with a different version of the gene, or a version of the gene that will encode a protein that has some sort of tag that we can follow, whether it's fluorescent or biochemical. There's all kinds of tools that are all kinds of experimental systems that become possible with this sort of technology that just weren't before. So it's really great. The moral part of this, that is what we really want to talk about, is the fact that we can also, in principle, use this technology to edit human genomes in human embryos that are then going to develop into people. So this is editing the germline. In other words, we can make genetic modifications in human embryos that will then be passed on if that embryo develops into a person.
And then has children.
So this was occasioned by, there's been sort of a flurry of papers and conversation about this idea of editing the human germline, because pretty much as soon as this technology was developed and understood, it was clear that editing the human germline would be possible. And so people sort of thought, oh crap, now we have this is going to become possible. Should we do it? Why or why not? And what are the implications of that? And then recently, a paper was published. So I'll just talk about the paper. So this is a paper published, I don't see the month on here, but just a couple months ago, in May, in Protein and Cell. And the article is called CRISPR-Cas9 Mediated Gene Editing in Human Tripronuclear Zygotes. And the first author is Liang, and the last author is Zhu. So this is a Chinese group. And they basically demonstrated that you could use CRISPR-Cas on human embryos. And they used these tripronuclear embryos. So these are some small percentage of human zygotes that are created through in vitro fertilization actually have 3 nuclei. So they basically get 2 sperm hit 1 egg, and so they have 3 copies of each chromosome instead of 2. And these embryos develop into blastocysts, but not much further. They basically terminate development. And so the idea of using this set of embryos, the idea was these are never going to develop into people, but they wanted to use this as demonstrating the system. And they basically showed that they could do it. It wasn't super efficient.
At some level of efficiency.
I mean,
there were errors, so it wasn't super precise or as precise as they wanted it to be.
So it didn't always hit the target. It definitely had a bunch of off-target cuts. So the enzyme that cuts DNA is directed by a small RNA that base pairs with DNA. So essentially, it directs an enzyme to a DNA spot. But if there's some mismatch, or that sequence, unbeknownst to you, exists somewhere else in the genome, the enzyme can go somewhere where you didn't intend and cut. So when they checked through to see if there were any other cuts, they had multiple off-target cuts. They had chimerism, where they injected the enzyme and the RNA into the embryos, but it didn't get all of the different DNA sites. So theoretically,
so
there should be 3 copies, 3 sites. And then when they looked at the embryo as it developed, there were some cells that had no cut at any of the sites, suggesting that maybe like the DNA is dividing too quickly for all the sites to be accessed before development continues, which I thought was kind of interesting.
Right.
Yeah, and also these are sort of abnormal because they have 3 copies instead of 2, so maybe the efficiency would be slightly better if there were only 2 copies. But these off-target effects are a little bit troublesome, because if you're trying to edit one gene and you actually edit other genes that are important, you could cause you could cause worse problems than you were originally intended. And we should also say that the initial interest in this sort of genome editing in humans would be to like remove sickle cell genes or to remove cystic fibrosis genes and replace it with a good copy or things of that nature. But the same technology, if it were used in that way, it would not be terribly hard to then move on to,
like
changing hair color or
I'm kind of
things of that.
short.
So that's
I'd
what
like to give my
people
kid a run at the NBA.
Right.
I'm just going to insert a couple more genes to get him a little taller.
Yeah, why not? Why not?
Why
So
not?
that's the moral, or that's the thing that people are concerned about. And I think the interesting thing to me is that a lot of the commentary around this paper, and this paper was fairly controversial. People were saying it shouldn't have been published. The authors claimed that they submitted it to Nature and Science, but it wasn't published there because of the controversy. I don't think that's true. I think it's
not a good
not that
paper.
great a paper. So I just think Nature and Science
It's an
didn't want
okay
it 'cause it's-
paper. Like it's not.
Yeah.
It's just not spectacular. The only thing that makes
not
it
spectacular.
spectacular is it's on humans.
Is the controversy.
right?
I would say
That's-
the
that's it.
novelty of it would make it more I mean, I think that I definitely think that they were rejected on a scientific basis, but Nature and Science have published Nature and
Yes.
Science have published
It's true. They do have a long track
scientific less rigorous studies off of the novelty and the controversy
That's true.
that they
That's true.
provide. So
Fair enough.
to say that there's some sort of like standard that all Nature Cell Science papers meet is a lie, and sometimes things get published just because they're controversial, like arsenic bacteria.
Right, or
Fair
the
point.
induced pluripotent
The acid
stem
wash
cell,
genes?
right?
Yeah.
Exactly. Yeah,
Okay,
so I
so-
do. So okay, sorry. Go ahead because I wanna play off something you're about to say. Actually,
Matt and I have talked about this previously, so he
Okay.
knows what I'm going to say. So
Yeah.
a lot of the commentary around this article talking about the controversy have said, see, there's all these off-target effects. This isn't ready. See, they're not super efficient. We shouldn't start using this on human embryos because the technology isn't good enough. And the problem with that sort of line of argument, though true, I mean, I think it's definitely true that we should not start doing designer babies with this technology at this stage. But the problem is that sidesteps the actual ethical argument because this technology is getting better very rapidly. Already, within a year or two, it has improved remarkably. The specificity is going to get better. The off-target effects are going to get lower. The efficiency is going to get higher. So we are going to get to a point where those concerns are no longer valid.
Maybe.
But there's still going to be the problem of, do we edit the human germline? Because we're going to get to a point where we can. And so the question is, given that this technology exists and is going to be able to edit the human germline in an efficient way, not now, but I would say within a decade at most, probably in less time.
see,
Given
okay.
that, what should we do?
Right. So I'm gonna sort of reject the fundamentals of that argument, but I'm going to accept the fact that the ethics of it are still something that absolutely need to be discussed. I actually don't believe that this technology is going to get us to the place where you're saying that it's going to get us.
Oh, thank goodness. Me too, Matt.
Yeah, so I think that this technology is phenomenal, and it's especially phenomenal in a research setting where what you can do is you can take a litter of mice, and you had just tried to use this system on a whole series of blastocysts. You did IVF, and so you get a litter of pups. So let's say you get 8 pups out of the mouse. And the first thing that you do when you go through those pups is you have to go through, take a small amount of blood, however you do your genotyping, and see what actually happened. Right? Even as precise as this has become, and we are starting to get animals in the first generation, which is phenomenal. We've never seen that before. There's a huge failure rate. And the problem with this system for me is that the front end of the system, the excising of a piece of target DNA, I think you're absolutely right. You could get to the efficiency that you're looking for as far as humans. Because, right, what efficiency are we going to accept in humans? So let's assume that you do this, right? You have Tay-Sachs. There's a chance that you're going to pass down Tay-Sachs, and that is a huge problem. You have death by age 4. And the alternative is that they choose to terminate the pregnancy. So this is a really high risk, high reward use application of this technology. Let's assume that the technology is 95% efficient. I don't think that efficiency would be accepted. And I don't. In the case of Tay-Sachs, if I was a parent, I don't know. I don't know.
But
it's really
you can genetically screen
but
the
you can
embryos
screen the
before you
embryos.
implant them.
Sure.
So you don't need 100%. If IVF is already you do IVF, you fertilize 10 or 20 embryos.
Right.
I think actually they've reduced the number that they do, but you are fertilizing more eggs than you actually implant.
Right.
So by a large number, right? And I think that efficiency I don't think is a problem.
I don't think that you're going to. I think the excision is good. Right. I think the excision part of this technique might get there to a point where you're comfortable that you can knock out a gene with enough efficiency where if you screen 20 embryos, you'll be able to pull it. But I think the homologous base repair, the natural stuff that you rely on in order to get your new target into the genome.
So this is not just a knockout, but a knock-in.
Exactly. You can't boost that efficiency past where the biology allows it to be. Right. And so I think for that piece of it, it's going to be really hard to get it to the efficiency where you're really comfortable that homologous recombination event hasn't been accompanied by other things that you didn't see. Now, you could make the argument that in the screening, you could do shotgun sequencing and, maybe that's the answer. Maybe for every one of these things, you get a high fidelity full genome screen. And if you have a high throughput, then maybe, but I think that this probably is the technology that I don't think this is the one that gets us there.
But I think the other problem with that too is the issue that I didn't even think of before I read the paper was that what if you get chimerism where you have a complete knockout of some cells in the embryo but not all of them. So it develops and for whatever reason you just don't get that full unless you start
if you're looking at cystic fibrosis or Tay-Sachs, you only need some of the cells to have one copy of the good gene.
Maybe.
Like cystic fibrosis, there are heterozygous CF people that don't have CF. That's a normal thing.
Yeah, but not in all cases will a chimera resolve the issue.
That's true. So, but again, I think that first of all, the idea that the technology may or may not get us to that point, this technology, CRISPR-Cas, I think the progress that has been made in just the last year, this technology, I think your point about failure rate and all of that, totally true. But that's also with now hundreds of labs, thousands of labs adopting this technology for the first time, starting to get good at it. And there are people who all they're doing is trying to improve efficiency.
I mean, I worked above the guy that basically, started implementing this stuff, and he was real good. I guess my argument isn't that it's not going to get a lot better. I just don't know that you're ever going to get to. I think that there are biological ceilings here that might be difficult to overcome unless you had just a series of screening protocols that we currently don't have, which maybe we will. But regardless, as I mentioned at the beginning, whether this technology gets us there or not, I do believe that at some point somebody will have the technology to be able to edit, and so it's still worth having the conversation.
Even if it's not CRISPR-Cas, I think that day is not far off, where we have this ability. We can do it efficiently. We can do it with a low error rate, with an acceptable error rate. So then, given that, what are the implications morally? So forget the technology specifics. Let's talk about what we're going to be able to edit human embryos.
So
within our lifetimes. I am comfortable making that prediction.
I think that's not unreasonable.
Maybe not by the time I'm having kids and you're having kids, but by the time our kids are having kids, we're going to be able to do this.
So my thoughts on this human embryo thing. So I think it's pretty clear, the benefits. So I think there should just kind of be cost-benefit analysis of this. I think the benefits can be really clear. If you have a disease in which your children will, or if you have the ability to pass on a disease that's going to lead to early death of your children, or you have a Huntington's allele or something like that, where it's this very clear-cut, your life is going to be shortened, your quality of life will be abysmal if this is not repaired. The idea of modifying a human embryo seems yes, we should do that. We should screen out, we should help people, or
Can I interrupt you for
Yeah.
just a moment, though? So, but for something like that, I feel like we don't even need to have the editing argument, because you can, pretty soon, we're just going to be able to do high throughput sequencing of embryos, IVF, and say like, oh, you want to make sure your kid doesn't have CF? We'll just pick the embryo that doesn't have the allele.
Well,
like
say that
that's
you have
going to be
like
way easier than
say that
editing.
you have like a homozygous dominant disease. So any child that's going to be genetically yours will have some sort of life-altering disease that you cannot prevent them getting.
Do those exist?
Can-
Because if you're homozygous for like people that have Huntington's disease, I think that's a dominant allele, but I don't think there are homozygotes of that for the most part. Most of the time,
You would
it's
be very-
one parent that gets one. You get one allele.
Yeah.
So in those cases, you have.
Let's just, for argument's sake, say that there is no way. I mean, because of what you're saying is like we don't need to modify embryos because we can just screen them.
For disease, I think that's the case.
So
I think for most diseases, CF, Huntington's, Tay-Sachs, we can just screen.
Yeah. So let's just say that there's like the only way to like save your child from having this debilitating disease is to genetically modify your embryos so that they are now cured.
Okay.
I think that's like very obvious. Like, of course we're going to do that. We kind of already do that, like you say, with screening. But now I think the issue is you have an embryo, say you have IVF, you have an embryo, and you want to make sure your child isn't blind or isn't deaf, because maybe you're blind or deaf and you don't like being blind or deaf or both.
Or the other way, and you are deaf and you want a child that's deaf because the deaf community finds that to be very important in forming that community.
I was going to say, like, I think then now when we're looking at the society we're making, we're like now making this judgment that like to be blind or deaf, even though you can have a normal life expectancy, is the wrong way to live, and that these sorts of like differences in ability should no longer be tolerated. And so like now we're going to make sure that like our society is no longer blind, is no longer deaf, no longer has like some sort of limb issue or some sort of like nerve issue where maybe you can't like lift things very well. So you want to make sure that your child has like very strong bones. And I think you start to make a society that is shunning people who are in it. Like you make a more exclusive, a more exclusive society. And I think that's the part where I'm just like, no, I don't even want to touch this. Like, if the most debilitating things can already be accomplished by screening, why are we going down this road?
Right. So I'd like to make... Oh, go ahead, Kevin, or Kate.
I was going to say, so I would like to know, because I agree with you, that slippery slope is super slippery, right?
It's a particularly slippery and steep slope.
I think so, because I think some of these things, like if you don't have any interaction with people who are differently abled than you would just automatically assume like, oh, I don't want my child to do that. I don't want that to be my child's experience. But there are people who are perfectly happy and enjoy their communities, especially something like the deaf community, that even though I know it's like very, there are people who go back and forth about like, should you be deaf? Should you choose it? Like the implants to stop.
Cochlear implants.
Yeah. To like make that sort of judgment as a society that like that's no longer acceptable, I think is quite a harsh and like
So and I think that the boundary between, and I think you articulated this quite well, since the boundary between legitimate disease like Tay-Sachs or cystic fibrosis and differently abled things like being deaf or blind or other traits, like what about being transgender? If we found a genetic signature of being transgender or gay, like the difference, like the line between those is a very fine gradient. I don't think there are bright lines. And so what principle, how do we develop a moral principle to say, given that this technology is going to exist, given that there are cases where we think it should be used, how do we then make decisions about when it shouldn't be used?
Well, obviously we just
I don't know the answer.
Ethan Hawke. GATACA. Yeah, I agree. Like where would the line be?
I'm not convinced that, and sometimes to a fault, I guess, I tend not to draw lines, or when I have to draw a line, I push it real far back. You mentioned that screening is going to become more and more prevalent as we have the ability to make sure that really debilitating disease sort of stops emerging into the human population. I'm going to make a historical argument, which I often never do because I often hate historical arguments, but we we've tried to do this in the past in much technologically worse ways, unfortunately. Like, we have tried as a species to identify people that we feel are inferior or not contributing well to the human race, and that's called eugenics. And in the couple times, at least in modern history, that we've done that, it's gone terribly for exactly the reasons that you're suggesting now. As soon as we felt like we had the power to decide who's sick, who's not sick, who's going to be contributing, who's worth having around, it all starts to fall apart, right? Humans historically have not been good at identifying who is and is not going to be useful to society or how to make society better. We're real bad at it. And I think
I agree with that,
even
but
though
I
we
think
can
it's a
do
different moral decision here. And I think it's different because previous eugenics attempts were basically removing agency from conscious creatures. So you're basically saying, like, this class of people is not allowed to reproduce. That was the way to do it. What we're saying now is that we are going to give people the ability to select before a child is born.
Right, but what I'm saying is
before there is any consciousness. And I think, morally, like, people looking back, because of the way that morality changes, you look back on the morals of a previous age and they're often abhorrent. And I think that's reasonable. So it is. I fully accept that somebody listening to me talk, if this record sticks around on the internet, and people look at this in 100 years, I might sound like a complete asshole. But I really do think there's a difference. Maybe just now I sound like a complete asshole. Fine.
I couldn't help it.
No, it's totally fine. But I think that whether or not it is right, I think that people will see it differently.
Right. But what I'm saying is, in both cases, even though we can distinguish a moral boundary here potentially between, sterilizing somebody, as was the case in the 30s and 40s, and potentially just modifying an embryo, what I'm saying in both cases, on top of that program, we had people trying to make the decisions of where those lines were drawn, right? And so not only has it been problematic in the way that the eugenics programs were carried out, but I'm saying the decision-making process of who needs to be a part of that and what situations it needs to be applied in were equally problematic. Right. And those things don't change. Right. We still have to make the decision as to, as Kate said, if you're deaf and you're blind. Right. Is that something that falls under that category? And I think that that's the same question that people had to deal with when we were in the 30s and 40s. Could you sterilize a deaf person or a blind person? Now, obviously, that's a worse scenario because you're doing it to an actively living conscious person. But the same question at the base is there. And I just don't think that we've done a good job of that historically. I think we will continue to do a bad job of it.
That's fair. But do you agree that there are situations where it is like let's take Tay-Sachs as an example. Because I think that's a useful extreme. I don't think there are many people that would be morally opposed to either editing that embryo or if someone has that allele, if a couple both have the allele, to say we're going to screen the embryos and make sure that we don't have a child that's born that's going to suffer and die before they're 5.
I would say absolutely I'm on board with the screening, and I'm not yet convinced that I'm morally okay with modifying the embryo to even in the extreme I have to take that argument because I can't yet distinguish where that line would fall, and so I'm not comfortable yet drawing that line.
But if the screening argument is acceptable for something like Tay-Sachs,
sure,
we know that at least part of something like homosexuality is genetic. It's not entirely, but at least part of it. So, it's on the same continuum. In principle, you could screen for or against that trait because that's a sort of, and that's not just one allele or not. There's a continuum there, and so in principle, you could screen and enrich, or what's the opposite of enrich? Deplete for particular traits. Things like deafness, blindness, homosexuality, transgenderism. I feel like all of those things, it's a line, and I think it's important, and I don't think we're on a 1 hour podcast, we've actually gone over an hour. I don't think on a podcast we're going to figure out the answer, but I think it's an important question that we need to know, given that either genome editing or genome screening technology exists and can be used or will eventually be able to be used to make decisions about human birth, how do we draw the line? And given that there are situations where I think most people would agree that it's appropriate, how do we draw the line where it's not appropriate? And I don't know the answer. It's a really hard question, but I think it's important that we have the conversation.
Yeah, I mean, I agree. I think my hesitance around the idea of actively editing just comes from the fact that I think you fall down that slope a lot faster when you're actively taking the approach to directly editing. You're certainly correct in that by my suggesting that screening is okay and editing is not, I'm sidestepping a problem that still exists. Clearly, I'm for being able to screen for something devastating like Huntington's and Tay-Sachs. As far as the rights of the parents to make choices as far as, what their child is going to have or not have, I'm not sure.
Yeah.
I also feel like one of the, even though this might be a silly argument, with screening, everything that is passed down is still natural. Like you haven't modified anything. So you still are just passing on your genes. This could be happening by chance if you just were to conceive naturally. Whereas if you've genetically modified your embryo and you have that child, now that child is going to have children who are also modified, and no one, as much, I feel like you are, the entire line of children, has not consented to that modification. You have one decision, and I understand, no one really consents to their genetic material. You're just born with what you're born with. But the idea now that maybe a couple years down the line, I guess you now have seeded a line of people who have this modification that makes them, I would argue, very different.
Yeah, but I think that that naturalistic argument where something that is natural is somehow more, it somehow has less moral agency, or it's less of a moral decision to select among natural options. This is maybe getting a little too far afield, but I have this argument with people about genetically modified food all the time.
I feel very differently about my food than I do about my
Fair enough,
children.
But I think it's a similar the sort of. I think that the aversion to editing that you have of people is not so different than the aversion that some people have to editing food. I think that the impulse is the same.
But
I think it's a different moral question.
No. But it actually is a little bit different than the food, because the food can't consent to being genetically modified. Whereas perhaps with a genetic modification, there's some side effect that no one knows until 3 generations out. And so now you have people who could have never consented to receive that modification now have to reap the downside of having it.
But you could modify them, Kate.
Unlike our food,
you could just keep modifying it every generation.
Unlike our modified food, our modified food is, tested for generations and generations and generations, and is the most thoroughly researched thing that has ever been put into your body.
Sure, but I think that the impulse.
I think the possible downside, the unforeseen effects of the modification that might bear out over generations. I do think that if you as a human have created it with your own hands, this problem, that seems more of a reason to me to not do it, even though I understand that there are naturally occurring genes that could, generations down the line, lead to some increase of cancer in your child. I wouldn't say that you're at fault, but if you actually did it and you actually did the modification, then I don't know.
And I think it's worth pointing out that a whole lot of scientists agree with exactly what you just said. So there was an open letter to Nature, basically signed by a whole bunch of people, expressing exactly what you just said. Essentially what they said was, we know that this is going to happen at some point. The ability to do this is going to happen at some point, but at this point, we just don't understand what the ramifications are down the line in multiple generations. And as a result, there's a whole lot of people that are calling to sort of put this thing in human systems on hold until we have exactly the discussion we're having right now on a more broad scientific and layperson scale too. I think this is one of those issues where this is not a science issue, or it's not restricted to science. Certainly scientists can inform what we view as, the scientific approaches and how accurate and all of those things, but I think that this is a much larger societal question that needs to be addressed.
You don't need to be a biologist in order to understand the moral and ethical implications around this question.
And there's so much science fiction written, I think, around this sort of idea that...
I honestly think Gattaca didn't do well in theaters, but it's one of my favorite movies, and I think very prescient, and I think is worth watching again.
Yeah.
Because I think that the issues there are also important issues. Like, how do we regard people? Let's say we decide that it is okay to do genome editing. How do we treat those people that have been edited?
Like there's going to be so many subreddits if that happened, like gen mods, like talking about their cool like genetic modifications, and like
I think
no one understands my genetically modified fedora. Oh, if only I could get a girl who doesn't like bad guys.
This is why I quit Reddit.
Another interesting implication is going to be when we can do this by choice as adults. So when we have like viral vectors where we can edit people that can consent, that can make choices.
Well, I think that's a completely different topic. I think that's actually, I want to hit on that too before we ended today, was that what we're talking about with editing embryos, we're editing the germline. So you're making an edit that's going to be present in all of the progeny throughout generations of that embryo. But now somatic mutations, so editing a full-grown human, you're just editing the cells that make up like a tissue, like you go... these viral vectors, you infect T cells, the T cells become resistant to diseases, or they gain the ability to fight certain diseases. Those modifications are not passed down. Those will only affect the person in their own lifetime. And I think the advances that are happening, so that's the somatic cell. So the advances that are happening in somatic gene therapy are really amazing. And I think those are going to come into fruition much sooner than the human embryo editing. And we'll see that a lot more. And I think like the somatic gene therapy, like that's just someone's choice. You want to get a viral vector that's going to make you resistant to HIV, you go get that vector. You go be resistant to HIV for 5 to 10 years until it stops working, and then you no longer are resistant to HIV.
But
little A&B jab.
Let's say we come up with a somatic editing technique to work on Tay-Sachs. Mm-hmm. I think cystic fibrosis is probably going to be easier because it's in the lungs, but let's just say we found something for Tay-Sachs. You couldn't give that. That child would not be able to give informed consent to that treatment.
Well,
how
it's
different
a child.
is that?
We have legal issues for that. We just have the parents say, knock yourself out. I make legal consent for this child. I don't want my child to die in 2 years. Please.
So why is that different than giving consent for embryo modification?
Because embryo modifications, it's not just that child that's affected. It's that child and that child's children and their children and their children and their children.
But by the time that child is going to have children, as Matt sort of mentioned offhand, they're going to be able to edit their genome back if they want to.
Maybe, but they're going to have that genome in every single cell of their body, and modifying one subset of cells in a very controlled way, like your T cells, where you just, essentially take blood out and put blood back in, is very different than modifying all the cells in your bone marrow and your toes and your lungs and your guts. Like it's a much bigger problem.
I think it's a bigger problem, but I think it's a difference in scale, not a difference in kind. That's the issue that I have, is that I think I want to know what the principles are, because I think that, we can make rules around particular use cases. And I think particular use cases are a little bit easier, or they feel easier to say, like, oh, this is like T cells, clearly okay, because we're editing one cell type, we're doing it in an adult, they can have informed consent.
But it's not only that. It's not only that. It's safer. It's much safer. We know the risks associated. We do infections in a clean space. We screen to make sure those cells aren't going to become cancerous. We make sure that they're alive. We put them in. There shouldn't be any adverse effects. Like, it's a much different case for the T cells.
Let's say that when I'm editing an embryo, all I'm doing is I'm replacing the Tay-Sachs gene with the wild type allele. I'm doing full genome sequencing of the entire embryo to screen to make sure that no other edits have been made. We know enough about molecular biology that we can say, I'm just changing this to a wild type allele. There are no other changes. We can do the screening. We can be super safe. I think it's a difference in kind. And I think, again, it's saying that the technology to do one thing is different and safer, and therefore it's okay, I think sidesteps the moral question, because there are other things that are more complicated that at this point, maybe it's not an issue, but I think it's going to be an issue in the not too distant future. And I think we need a set of principles around what, as a society, what we're willing to accept and what we're willing to do that is based on principles and not specific use cases, right?
Yeah. I mean, we have done a poor job of that previously. I think making these sort of principled lines, because unfortunately we aren't great at seeing into the future as to where those lines will shift.
And technology moves too fast.
Right, exactly. So I don't know, maybe for now the specific use case is the best that we can come up with. And, as these things develop and as we start to understand what this technology is or is not going to be capable of in the future, maybe we don't and can't have the clarity right now to establish those principled lines.
Maybe. I think that's unfortunate, because I think that doing it one by one as these things come up, I think we're going to make more mistakes if we do it that way.
Yeah, maybe. I still like the plastic surgery idea. Just CRISPR-Cas as a plastic surgery agent seems awesome.
Inject it into your tailbone and
exactly.
make a
Are
tail.
you trying CRISPR-Cas like boob jobs?
Yeah.
Yeah.
Okay.
I think that would be awesome.
That's a whole new scale. That's a whole new scale of spam mail that's going to start coming.
So I think we should probably make mention that there's currently no funding going to anyone who tries to modify human embryos. So like in the United States, in the United States, this is not research that's actively being pursued.
Right.
However, some news articles have done some research and found that there are at least 4 other groups in China who are doing research like this. So
did that surprise anybody? That didn't surprise me in the least.
It didn't surprise me. I don't know what that says about me and how I view China, but it didn't surprise me that much.
Yeah, I suspect that there are more than that.
Yeah.
And well, the nature of scientific research means that wherever this research is being done, the implications are going to hit all of us.
Yeah,
so we need to have these conversations.
Definitely, no question.
So anyway, does anybody else want to say anything?
Oh man, I have so many things, but I think I'm probably just going to bring up this small news article that I read, that when Kim Kardashian got her IVF,
oh God,
the reason why she knows she's having a boy is that she only implanted male embryos.
Oh my God.
I know, interesting, right? Kim Kardashian, right into like our news, right on the cutting edge of science.
This is why we keep you around, Kate. I would never ever have come across that, and that's really
well, apparently
interesting.
you do not read Jezebel, my friend.
Apparently not.
that is true. That is accurate.
But I also own her book Selfish. It's an excellent book, but don't buy it if you're under 18 because there are naked pictures in it. So I'm not going to endorse it for you children. Wait until you're
We already have to have the tag, I think, on this iTunes. I think we
yeah, I think Kardashian needs to be a tag for episodes. Episodes where
yeah.
Anyway, so I guess we can wrap up there.
Yeah.
This has been Audiommunity, episode 13, recorded on, what is the day today?
I think it's
it
the
is
27th.
June 27th, 2015. I'm Kevin Bonham with Matt Woodruff and Kate Franz. And you can find us, I should really have something up here about the outro. But Audiommunity is found at emunity.org. It's EMMUNITY.org. And the music at the beginning and the end of the podcast is performed by Rachel Reineck. We'll see you next time. Bye.
Woo.
Cool.
That was heavy.
Yeah.
Yeah.
It's good discussion, though.
That was good. That was really good.